Okay, I'm going to start now. Today is Monday. And in last week, I think everybody like in Asia, America, suffering a lot. Well, so I still like very grateful for our school. Our school president. I'm from Wednesday University. And this is my oldest building of our school. He cited a sentence from Martin Luther King, say that if there's one place there's injustice, there's injustice everywhere. And further, our president said that if there's one violence against one group, there's a violence against everyone, every community. I'm very grateful. Yes. Well, I kind of like a feeling like my emotion, like my mood is coming back. I will try this again. Okay, let me start. This is my slides. And let me see my pointer. Yes. This is I learned from our like it's a journal club or seminar. Yes, it's from seminar. I learned this. Okay. Today's the number 6 lecture. And this is a focus. So this chapter is focused on proton pump rhodopsins. Major focus on proton, and they're quite very interesting several parts. One thing is the VIR, and this is this is super new to me, because it's like a virus that can just like virus also have eye. A virus can express, but to me, it's kind of like, of course, virus have a membrane. There's a protein on the membrane. Of course, it's possible based on the concept so that like rhodopsin, this transmembrane protein, can express on the virus, but it's still pretty new. Like to me, it's like how wow. The virus can also express rhodopsin and the response to light. And this is what I feel it's really, really excited to me. And the second part of this chapter, a detailed focused and like talking about the several types of like proton pump. And we can see here from this image, like actually like some groups is a channel rhodopsin, like those are channels, those are ion channels or cation channels. Like we can see like ICR as the channels, and the CHR, CHR1, CHR2, VCHR, that's cation channels, but majority is a pump, either like a proton pump and chloride pump. And in previous lecture, we already talked about that. Some just like 1 point mutation at the asparagine 85, then can convert a proton pump to chloride pump. That's one single amino acid mutation. So these two groups, proton pump, chloride pump, is actually quite similar. And the third part is research like molecular engineering that can convert an arch to a voltage sensor. I think that's quite also excited. Because previous we know the voltage sensor is genetic voltage sensor or some chemicals that binding inserting into the membrane. then can sense the voltage change inside the cytosol. But in this study that I think it's published in 2000, very recently, very recent. If it's published, I think it's a 2020. If it's not, then 2016. We will check later. Anyway, it's a voltage sensor. That's and based on rhodopsin. I think that's a super cool. And other part of this figure show that because there's some rhodopsins can bind to some like very complicated molecules, and those bindings can change like some characteristic and property of the channel rhodopsin. So in human beings, those binding like G protein coupled receptor, those rhodopsins are type 2, and the rest of them that's from the microbia, that's type 1. 1 minute. Okay. So rhodopsin, we know that this name is coming from rose and the Vaginaceae. That's rose because when we isolate the tissue and looks like a pinky color, and isolate like the photoreceptor, like all the segment discs, and those proteins like with a rosy color. But we know that the carotenoid or retina are yellow, like the carrot, are yellow. But when this ligand binding to the protein, the rhodopsins, these colors will change to a variety of different colors. So that's why for the rhodopsin in our human eyes, it became a rosy picture. But like in the bacteria, that's another color, that's a purple. And we can see here. And that's when isolated those bacteria, and the scientists find that there's some purple patch on the membrane of those bacteria. So this image is coming from this article published in 2018, and it showed that like after binding the retinol and the whole rhodopsin that combined with the pigment and we can still say the pigment, the ligand, the retinol, and the protein together, and it will change to another color. And for different proteins, different kinds of rhodopsins, and that color will also different. So in addition, it's not only like this rhodopsin that the color is changed. So when it absorbed a photon that activate excited by light, and this color will be also changed. So that's the best of the voltage sensor, the Archeron 2 basic concept. And we'll talk about that later. So this is the figure 1 of this chapter, and we can see that so this is a general figures of the general rhodopsin, and the all transretinal binding to the transmembrane helix number 7. This is 213, and after capture a photon, and it can change the conformation. And most of this, and this figure is quite similar from the previous one, but I still put this one on the slide because this slide it focus on like their functional, like some are like a sodium pump, some are chloride pump, and some are proton pump. And for generally we can see this image, and we can see that like a majority is a proton pump and a chloride pump. The first proton pump, the AR, and is found by German group Dr. Heckman in 2006. I think I can see someone. Yes, in 2006, quite early. And there are several different types, like bicurorhodopsin and heterodopsin from archaea, and then there are proton pump and chloride pump, and in fungi and algae and green algae, and this shows proto. I think this is DTD. Oh yeah, it's a proton pump. So it's a DTD motif. That's the RG proton pump. Also, the DTG motif bacterial proton pump and the pentameric proton pump, chloride pump, and in the, so this is the first like very exciting point of this chapter. That's the giant virus. That's the VIR. So these slides just show that's from the heterodopsin, from the bacteria. I see like some purple patch on the membrane, and when I isolate those patches, then we can see that this bacterodopsin that forms a trimer, and together then form the patch on the membrane. So we can also see that because like in human eyes on the discs of the segment of the photoreceptor, they also forms patch like on the membrane. So when we do experiment, we also find that some like ACR, especially ACR, like GT-ACR, that really forms patch on the cell membrane, and that's not. I think that's determined by the nature, like their the molecular like properties that they form this patch. It's not quite be considered like toxicity. But if like those rhodopsins, if like form like clusters in the ER, like in the intracellular part, I guess maybe that have some cytosol like toxicity effect. But here, I think they're pretty normal. So in this, we're focused on the bacterial rhodopsin. So bacterial rhodopsins. So just in last slide, in last lecture, we're talking about that like the human those type 2 rhodopsins, only by like the 11-cis retinol. Then after capture the photon, then change conformation to the all trans retinal. But the bicurable rhodopsin, if that side empty, it can bind both either 13-cis retinal or all trans retinal. That both ways works. Like in the dark adaptation, it will form like 50-50 ratio to binding all trans retinal or 13-cis retinal. And under the light, the all trans retinal will decrease and 13-cis retinal will all trans retinal increase and the 13-cis retinal decrease. And that means binding. And because like for bacterial rhodopsin, they binding all trans retinal, and a conformation change will be captured by a photon, and it will change to 13-cis retinal. So the binding of all trans retinal will be increased. And here and a further topic of this chapter, I focused on this and several steps of this protein that like binding the photon, then change the conformation, and and it caused like some conformation change on the proton switch from one side to another side, from a donor and like some to bind into the Schiffer base, then Schiffer base like deliver this proton to another residue, then goes to extracellular site that pump the proton out of the cell. So this is a further marker with the different steps. We remember that, and I mentioned, and let me see, it did, yes, it marked it here. So see, so at every step of this conformation, and the light wavelengths, optimal like absorption light wavelengths were different. Like in resting state, and its optimal excitation wavelength is 570, and like in the open state, is 410. So when we see, so would you mind that go back to very early? So what I'm talking about is this image, is that so when capture with the photon as a conformation change, so every conformation change, the color of the optimal absorbing color of this rhodopsins is changed. So we just talk about, let me see where it is. Yes, here. See, like in open state, it's a quite blue shift, and the resting state, it's a 570. I think it should be like orange color, like quite orange. Like, and this is also, I think it's already like very close to like ultraviolet. Yeah. And there's also internal water molecules always stay in these critical areas, and that's very close to the Schiffer base of the retinal. So of the water, and it forms a very localized like watery um, like a small compartment at this, and forms a hydrogen bond network at this area, then to control the conformation and open or close state of this proton pump. And most importantly, another aspect is that it is determined like the absorptance wavelength of this rhodopsin. So the most important to determine like the color, the absorption color, is this 19-3, the leucine site. Okay, then let's go back to here. So for a proton, so at this L to M1 stage, and a proton were like will be delivered from Schiffer base to the asparagus 85. We remember that that's the critical site to determine its proton pump or chloride pump, like 85. And then the next state, M1, M2, it's considered to be like one proton will be pumping out into the extracellular site. And the M2 and the N, and at this step, a proton was shipped from this asparagus 96 to the protonized Schiffer base. And at the stage of N to O, and there's a proton that coming from the cytoplasmic side, then binding to the asparagine 96. And at this asparagine 85, and with lysis 225, there's a tiny hydrophobic environment around here, and this block the way of like the proton shift away. And this mutation of lysis 96 to alien will drastically decelerate the deprotonization of a process of RSB. So now let's talking about something about this virus channel rhodopsin. And this virus channel rhodopsin is found in the green algae that very close to Hawaii. Yeah, Hawaii. And it's found that in the green algae, there's some like a giant virus. And when you see that this is a whole like the micro cell, and this is the size of the virus. This is a pretty big. It's about 200 nanometer. So to compare to other different kinds of the virus, like AAV, it's quite small. It's 22 nanometer, and this giant virus is about like 10 times larger in diameter than AAV. And Ebola, of course, it's a long line, and it's the largest. And this year, the SARS virus is about 100 nanometer. The SARS or the coronavirus, and its dimension is about 100 nanometer. And this giant virus that infect the green algae are like 2 times like larger in diameter comparing to coronavirus particle. And this rhodopsin is like it forms like a 5 subunit, and like those ions were coming through the central pore of this rhodopsin. So in detail to check like each like subunit, and we think is also 7 transmembrane. So we can consider that it's kind of like 5 of these rhodopsins that forms a complex, and the ions, cations were flux through like in the central pore of these 5 rhodopsin. And this study is summarized in last year. So the last part of this chapter is actual. It's a fluorescent voltage sensor. So at a different pH, and the sensor were different, and the protons flow, and the proton flow like directions were also different. And this study is coming from Peter Hagman. And remember that the first on AR is discovered in 2006 is also from Peter Hagman's lab. And this, yes, I'm correct. So this voltage sensor were published in 2020, at very recently. An arch is here, and let's say the arch is I'm consider it's best on CSR rather arch 3. An arch is a proton pump that pumping proton that's from intracellular to extracellular. And for molecular engineering, Peter Hagman's lab that mutate this 96 site. This is because if they mutate this site, then it has no photocurrent. This means that so after capture light, there's a conformation change and absorption wavelength will change. Of course, that's related because it's a proton pump, so it's quite related to the pH. But if if like the scientists only want it to be like a sensor, but don't want it to be like a pump to play a role in a change of the membrane potential, then it's better have no photocurrent for this sensor. So Peter Hageman lab that do mutation of this site, then it will totally like block the photocurrent. That means that the proton like transfer that within this rhodopsin. And we can see here, so at like a normal pH, pH 7.3, and the color, it shows a pinky color, and the proton or binding will flow in, and it's under a positive membrane potential. But at the pH 11, and it will switch to a yellow color, and the proton will flow inside into the cell, and it indicate a negative membrane potential. So, but we still need to focus on, because on this study, I go through this paper and discover that they do this research like at pH 8. So is it okay that it still can like works in HEK cells or other neural cell types? I think it's still need to for a study. Okay, thank you. That's all. Thank you.